If there's one big question that all long haulers want to know the answer to, apart from when will I get better, it's is viral persistence driving this bloody awful condition? There's a lot of circumstantial evidence, but we haven't yet found the smoking gun. So in this film, I chat to microbiologist and published long COVID researcher Dr. Amy Proal to get her take on the subject and why viral persistence, if established, could explain pretty much the whole thing. Okay, so since the beginning of the pandemic, we were looking at, since we sort of realized that long COVID was a thing, we were looking at four possible causes for what might be driving it. They were hyperactive or dysregulated immune response, autoimmunity, viral debris, or viral persistence. And more recently, we've added a fifth, which is reactivated latent viruses. Of that kind of five, it's probably not just going to be one of them. I think there's more than one of them going on. What's your take on the balance of that? Where are the hot parts of that five-piece puzzle for you?
Yes, Jez, it's funny because independently from you, I'm not sure if you saw that along with my colleague Mike Van Elziger, who is a neuroscientist at Harvard, we wrote a review article on biological factors that may contribute to long COVID. This was well over a year ago now.
Yeah.
And we actually came up with basically the same five factors that you mentioned, but we actually have some additional factors that we mentioned as well to include, which also are dysregulation or imbalance of host microbiome communities. In other words, imbalance of the collective ecosystems of organisms in and on us. And then we also honed in on two other topics, which were how some of the things you mentioned, possible persistence of the virus, reactivation of other viruses, for example, Epstein-Barr virus reactivation in patients with long COVID could impact signaling of the vagus nerve to the brainstem. And then we also mentioned some of the potential coagulation or clotting issues that could occur. But to go back to your original question, let me think then. So you mentioned obviously immune dysfunction first.
Yeah.
That, of course, I strongly suspect that obviously the immune system is getting caught up in everything that's going on with long COVID, no matter what. But the question to me as a microbiologist is why? So part of what is important to understand is that when you see, for example, and you measure cytokines in a blood sample and you see that there's activation of cytokines, you can assume that that means that the immune system in a sterile environment has activated and is expressing those cytokines just because there's some sort of aberrant signaling. Or you can also consider factors that activate cytokines, and that goes into the microbiome component that I'm interested in. Because one, as I explained before, we're seeded with trillions of interacting organisms. So bacteria, viruses, fungi, we have these communities, not just in the, I think people are familiar with the term gut microbiome, right? We have this ecosystem of organisms in the gut, but we also have vast ecosystems of organisms in the oral cavity, in the mouth, also in the lung. There's a microbiome. So you have, and most human tissue and blood now is no longer sterile. There are microbiome communities in tumors. There are organisms in so many human body sites. So what can happen is that under conditions of imbalance, perhaps when someone is infected with COVID, you will see leakage of some, first of all, you'll see emergence in those communities of organisms that can drive more inflammatory processes. So sometimes we refer to those organisms often as pathobionts. So the bacteria in your gut, they can be in a fairly stable, balanced state where they're connected to human signaling in a conducive fashion. Or under conditions of inflammation or imbalance, the organisms can actually change the genes that they express to start acting more as virulent organisms, where they start to express proteins and metabolites that actually disrupt our human signaling pathways. And so what can happen is when someone gets COVID, the organisms in the gut, the mouth, the lung, they start to turn towards this more virulent or pro-inflammatory state. And then that can affect the lining of the gut or the oral barrier and cause leakage of organism into the blood. And for example, that will stimulate a huge amount. Then the immune system in the blood recognizes infiltration of those organisms and may respond with all kinds of cytokine activation and activation of other immune cells. So I do think that there is immune dysregulation in patients with long COVID, but I hope that we can dig into the root causes of why and involve the microbiome in those analyses.
I haven't really heard too much discussion about. I've heard, there's been microbiome and gut microbiome's been a topic people have been talking about, and there's a decent suspicion that something's out of whack there with long haulers. But the idea of the microbiome elsewhere contributing is something that I've not heard much discussion of. I mean, is there any research out there that's looking into this? How many? I think this is, this is probably a niche that not many sort of clinicians or academics are maybe that in tune with.
There is some really compelling research on microbiome in complications with long-term symptoms with COVID. One of the studies that most caught my attention was a study done by Alessio Fasano at Harvard Mass General. And what they were looking at was children who had multi-system inflammatory syndrome. So that's a condition in which some children develop a very profound hyperinflammatory response several weeks after initial COVID disease. And the children are often asymptomatic or just have mild COVID infections to begin with and still get this very severe inflammatory syndrome a month or so later. So what Alessio's team showed is that he actually looked at the gut microbiomes of these patients. And the microbiome itself was not in great shape, the organisms that were already there. But a caveat was that SARS-CoV-2, and this goes into another topic you mentioned, the SARS-CoV-2 virus was still present in the guts of these children, somewhat as part of it had joined into that ecosystem, right? And so SARS-CoV-2 was still there in what we'll call a reservoir in the gut, in a low level amount in the gut. And what was happening is they measured a marker of gut permeability. They measured what's called zonulin. And they were able to show through that the gut lining was more permeable. And then the spike protein from the little SARS-CoV-2 that was left there was leaking into the blood. And that leakage of spike protein in the blood was what was driving this hyperactive immune response in children who were genetically susceptible. So that series of events is where you start to connect some of the topics, as you alluded to before, saying you could have a situation in which a patient doesn't fully clear SARS-CoV-2. It remains in a low-level reservoir, a little bit in the intestinal tissue, a little bit of virus remains in lung tissue, and then the background organisms in that body site, the ecosystems of organisms that were already there, it joins in a sense, and often even promotes imbalance of those organisms in a further fashion. And that's been studied with other respiratory viruses. For example, influenza. One of the teams that we're working with published a 2020 paper in Nature where they showed that when influenza virus infects the lung, the bacterial organisms in the lung shift towards a more imbalanced state, that the background microbiome and some of the existing bacterial organisms, for example, in their case, Pseudomonas, sort of popped up and became more virulent in those patients and then started to contribute to the disease process along with the effects of influenza. So you could have very similar trends happening with SARS-CoV-2.
With that study with the kids who had the leaky virus in the gut, that's not a particularly accurate way of describing it, but how far was that acute? Was that during their acute infection, or was this some way in?
Good question. So the virus was still measured in the guts of the children 3 or 4 weeks out. So 3 or 4 weeks after acute infection.
So they're still, by that time, they're PCR negative, right? So
definitely. I should make that clear that in that study, and that's a real trend to understand, is that there have been now a number of studies that have still found the genetic material or proteins created by SARS-CoV-2 in tissue samples, or in this case, in the gut microbiome environments of patients after acute disease of COVID. And nearly in every study, the patient tested negative via nasal swab testing when they still had the virus or the genetic material in the other body site. Yes.
So if we're looking for the sort of the body sites that might be harboring some sort of viral reservoir, which are the top 5 places that we'd like to look or would be sensible to look?
Good question. So intestinal tissue, might be my number one site. And that is because there is actually a fairly robust literature of viruses, including RNA viruses, persisting in intestinal tissue. It's a nice little niche. You can hang out. The immune system doesn't recognize you too well. It's not hard for the pathogen to get into the gut in the first place. The patient just has to keep swallowing. And we do already know that many patients with COVID and long COVID have GI symptoms. So it's not that hard a site to access. So certainly, and one of the things that has made me particularly interested in intestinal tissue is one of the researchers that I most respect in the MECFS research community, a scientist named Dr. John Chia. For a long time, he has been collecting intestinal tissue samples from patients with MECFS via endoscopy. And he has stained hundreds of those samples for enteroviruses, which are single-stranded RNA viruses that actually have a lot of similarity to SARS-CoV-2. They also can be acquired via respiratory infection. Often people who get an enterovirus infection only have mild or asymptomatic symptoms, but can develop chronic symptoms. So he has found this enterovirus reservoir in MECFS intestinal tissue and published that in at least 2 papers that I wish got more attention, to be honest. And that always set the stage for me to consider that intestinal tissue was a good site of persistence for other RNA viruses as well. And beyond that, the intestine is a lot of different body sites are innervated by the vagus nerve. And the vagus nerve is a very important nerve that connects to most major trunk organs, but it absolutely innervates the gut in a major capacity. And so what happens is if there's a bit of pathogen reservoir in the intestinal tissue of the gut, there will be some inflammation likely associated with that reservoir. The vagus nerve will sense that, and it conveys a pro-inflammatory signal to the brainstem, which is a region at the back of the brain. And one of the things that Mike and I delineate in our long COVID paper is how the brainstem contains these overlapping nerve bodies that control the feeling of being sick, the feeling malaise, flu-like symptoms. There's also a nerve body that controls autonomic symptoms. The ability to move from a sitting to standing position is regulated by neurons in the brainstem. There's also control of nausea. So all these overlapping areas in brainstem, if the vagus nerve sends a pro-inflammatory signal to that area, people develop sets of symptoms that are very similar to core symptoms we see in patients with long COVID and MECFS. So there's actually, as a takeaway there, a mechanism by which a little bit of virus in the gut can lead to signaling in the brain that even impacts symptoms that are not only gut symptoms, but autonomic symptoms, flu-like symptoms. So it really makes sense that when you start to see just what patients are experiencing, that gut reservoir is a possibility.
And also, it would potentially explain why we remain in this state of sympathetic activation the whole time, because the vagus nerve is going, we're being attacked, exactly. In a very simplistic sense.
Exactly. Yep.
Where else, apart from the gut, where else would you look?
Lung tissue. And by the way, we have 2 teams that are doing this. We're actually, we're writing some grants, although we are also looking for private funding to support the studies to move them quicker if we can. But we have another team that is now able to get lung tissue obtained via a bronchoscopy procedure. And we are going to work that tissue up very well because, or the teams involved are, because certainly it makes sense. The lining of the lung is another conducive. We know that SARS-CoV-2 enters the lung. It would be at this point a little ridiculous not to check and say, well, I know we have a respiratory virus that loves the lung. I wonder if the tissue of the lung would not harbor some of this virus in a persistent state. So that's the second area that I would check, would be lung tissue itself. Again, the vagus nerve does innervate the lung as well. So you can have that same kind of connection between body sites in the brain from persistence in the lung also. And then after that, to be honest, there are teams that have already looked in appendix tissue, hemorrhoid tissue, lymph node tissue, and I'm trying to think a couple other tissues like that. That seems like low-hanging fruit as well. The appendix sometimes, in simple terms, tries to purify what might end up in the gut. And so you might get some influx of virus there that gets contained. So tissues that are near to the gut, like appendix, and connected in that sense, might also make sense. And then also, to be fair, we definitely need good autopsy work where we look at tissue that cannot be collected from a living person, because I would love to check that the virus itself could not, in some patients at least, still be present in the vagus nerve itself or in the brainstem itself. And the reason that I say that is because there has been an autopsy study in ME/CFS in which they looked at the tissue of the brainstem in an autopsy study and found enterovirus genetic material, the RNA and protein, those same enteroviruses that were found in the gut, in the brainstem itself. And the vagus nerve there are some pretty interesting studies showing that the vagus nerve can be directly infected. In fact, I was just reading one from a team in Japan who found that the vagus nerve could serve as a conduit by being directly infected by influenza virus between the gut and the brainstem directly. And if you think about it, SARS-CoV-2 is a neurotrophic virus to a large extent. It seems to like infecting nerves. So it seems unlikely that it wouldn't infect the body's most central nerve in some capacity, the nerve that would allow it to travel throughout the body, the nerve that would give it access to the central nervous system. It seems possible. And last, there was a German autopsy study of acute COVID patients. And in acute COVID, they found SARS-CoV-2 in the vagus nerve and in the medulla oblongata of the brainstem. So it's possible. So it's another thing where I think we should check.
I mean, it's a bit scary, isn't it? The idea of it being in your central nervous system to that degree.
It's scary, but at the same time, we know how sick everyone is. So I personally am not sure if it's more scary to think that our immune systems are just failing us in some random way, which is scary too, or that there may just be a more concrete reason for some of these symptoms, which is infection of these sites, right? So I guess it's scary in a lot of senses, but yeah.
Why is the body not wiped out these reservoirs? I mean, we've had these huge immune responses with, our T cells and B cells, our antibodies have gone potty. How are these reservoirs managing to persist without being hit by the immune system, especially over a period of months or even years, right? If that is the case, if we are assuming viral persistence.
It could be. And so the answer would be that it seems like our immune systems are not doing so well in COVID, at least in a number of patients. So T cell responses are definitely impacted. They have been shown to be there's problems with T cell responses in a number of studies. In fact, there was even a study recently, and I think it might still be a preprint, but either way, which showed that perhaps SARS-CoV-2 can even directly infect T cells. So if that's the case, and T cell function drops down, that is well known in the literature. It's usually referred to as, lymphopenia. There's just other issues where T cells are no longer functioning. That is one of the circumstances in which pathogens do not fully clear. Without a robust T cell response, it's fairly easy, not easy, but there's ways for pathogens to stick around. And also SARS-CoV-2 creates at least 10 proteins that knock down host interferon signaling. So interferons are cytokines that are actually supposed to respond. They're the first line of response against most forms of viral infection, not just SARS-CoV-2, but other viruses. That gets into also how other viruses may reactivate. And so by disabling interferon signaling in patients in a number of ways, you also lose that part of the immune response to an extent that also plays a role in clearance. So there's a number of mechanisms by which the immune system may actually literally be hijacked by SARS-CoV-2 in a manner that's impacting and dysregulating T cell function, interferon signaling, and perhaps just antibody production in general in a manner that would make it easier for the virus to not fully clear.
I mean, it's interesting when we look back to the reactions to the first vaccination jab, and I did a study on this as well at the time,
Yeah.
and there was I mean, it was about the middle proportion were not basically not made better or worse, but there were some people who suddenly found their symptoms get a hell of a lot better, and there were other people who were made worse. So, I mean, it makes you wonder, right? Because those people who got better, are they getting better because suddenly it's given their immune system a kick that's cleared the virus? Or has it been like a sort of a reboot on their immune dysregulation that
Right.
has basically calmed down the immune dysregulation? And what's happening with the people who are getting worse? Well, they're probably basically a bunch more spike protein in their blood is creating more autonomic
Yes.
issues. So I mean, it really is a complex puzzle. And the idea that there is persistent virus might explain some of those people who did get suddenly better.
I agree. I thought it was a very interesting approach to start to study the impact of vaccination in patients with long COVID. There, as you mentioned, are many variables involved in how you would interpret responses because, yes, a new influx of antibody maybe could help clear persistent virus, but also once the virus has gotten into a reservoir where it's embedded itself and, in a sense, protected itself from the immune response, it's hard to know now if an antibody response that would have been possibly more robust during the earlier stages of infection can still reach it in a little reservoir. Because, for example, in post-Ebola syndrome, which is now a condition that's actually connected to chronic and latent persistence of Ebola, the Ebola virus persists in what that research community calls anatomical sanctuaries. So in the tissue of the eye, in semen, in breast milk, in other body sites that are, what are called immune privileged. And what that means is that the immune system does not penetrate those body sites as much as it does many other body sites. And so you could have that same trend here where SARS-CoV-2 is sneaking into these areas, which it would do, where it's increasingly protecting itself from the immune system. So even if you give the patient a new antibody influx, it might not find it. I don't know.
Well, I mean,
So
well,
there's
80% of people didn't get better with the vaccine. So
right.
there's the point, right?
I know.
um,
Yeah.
so let's say that we do a bunch of biopsies and we find we go, yes, there is persistent virus and this is where it's hiding. What the hell do we do about it?
Honestly, antivirals make a lot of sense to me. I think that it's exciting for I've been in this field of studying infection associated, what I call infection associated chronic disease, conditions initiated or exacerbated by infection for many years now. And one of the frustrating aspects has been there has been very little antiviral development. We know already, for example, that viruses like Epstein-Barr virus or the herpes viruses contribute now to a growing number of diseases that were considered to be purely autoimmune, like multiple sclerosis and lupus. And still, there really hasn't been much movement to just go ahead and really try to develop better antivirals. But now with COVID, a number of drug companies are creating SARS-CoV-2 antivirals. And I have been excited by that, although it's been a little hard to follow how those antivirals are even working in acute COVID. At this point in time, Paxlovid, for example, seems like a promising drug, but people are just starting to take Paxlovid for acute COVID. And I've basically been forced to track what's going on somewhat anecdotally. I'm not even sure who's collecting data. So there's, I guess, in a stepwise fashion, personally, I'd love to better understand how the antivirals for SARS-CoV-2 are working in acute COVID. But then as soon as we get an idea that they might be helping, it seems pretty logical to start to trial them in long COVID.
What research is there? I know you and your team are involved in trying to dig in some of these territories. Are you aware of any other research that's going on out there which is digging in these sort of hotspots?
You mean looking for virus in tissue
Yes.
samples?
Yes.
It's been interesting that we seem to be one of the few groups doing that. I do understand it's hard to get tissue samples. That's been something that I've been working on night and day. So it does involve more time. You have to be able to find the surgeons or physicians that can do the biopsy or surgical procedures to get the tissue. And one thing that's very important for the research teams that we're working with is we need to stabilize the samples immediately. So we need the RNA in the samples to be preserved right away, because if it degrades in any fashion, we may actually not find the genetic material of the virus because it degraded before we were able to analyze the sample, which would be a disaster. So we collect our samples into a special stabilizing solution for RNA. So we have to get those into the hands of the surgeons and the MDs, and they have to get the ethical permission to do the procedures. So we've been working that out. And I think that there are a number, it's a challenge. So I do see why teams don't take it on. And it's definitely easier to collect blood and do some analyses on that fluid. So I feel the teams that have been jumping in thus far have been doing a bit more of that. That being said, the NIH, through their Recover initiative, is apparently collecting tissue samples and doing autopsy work on patients with long COVID. And I've seen that they're doing that. And I've heard that they are as well. I just don't know how fast that research is moving forward.
What would it take to get more of this research happening?
Funding.
Is it that simple?
Yes, because when we're applying for government grants, we'll probably get funded at least a year from now. So you have to apply, your grant gets scored, you come back, this is about 8 months down the line, you revise it. It's a long timeline. Whereas right now, if we can at least just start and get the money to collect the sample, for example, just even get pilot funding to be able to go ahead and get 10 intestinal tissue samples, we can work those up, do all kinds of sequencing and molecular analyses to actually say, look, is there any signal here? And then when we apply for a government grant, we have what's called pilot data that will improve the process with that grant. So what we're trying to do is speed up the process by getting at least enough funding initially through private donors to get these studies started. I would have to say that that's the thing that is most important. Although obviously finding the surgeons and doctors who can collect the tissue and stabilize it correctly was the second biggest challenge, but we kind of nailed that one for now.
So to sort of summarize then, we've got a dysregulated immune system, which is kind of interesting, but probably not the main driver. We've got autoimmunity, extremely interesting. Autoimmunity may be connected to viral persistence, which is clearly also very interesting. Immune debris is probably a side effect, as are the latent reactivated viruses.
Definitely with the immune debris, it depends. I know that there are 1 or 2 teams that have found what they call viral fragments and monocytes or immune cells like that, but you have to consider that monocytes are cells that are trying to clean up byproducts of what has already been broken down. So it's certainly possible that parts of the virus or fragments of the virus would be found in certain immune cells like monocytes or in certain areas of the body, but that doesn't mean that there isn't just virus somewhere else that is still an issue. So the debris thing, to me, it's tough because one of the things with RNA is that you'll see teams, when people find the SARS-CoV-2, the backbone of SARS-CoV-2 RNA, they'll say, oh, that's just RNA, that might just be a fragment. That might not be the virus, but it's a strange argument because RNA is rapidly degraded. That's why we have to go through those great lengths to preserve our tissue samples. So at that point, you have to almost come up with a mechanism by which the enzymes called RNase enzymes in the body that every cell produces that break down RNA are not doing that, so that this fragment, if you will, is still in tissue, and it gets complicated. So the viral fragment debris stuff is not, to me, I think the virus is more likely itself, but yeah, I don't know.
I think that it's far more likely that the debris is active trash that's coming out of an active infection. Exactly. Because if the active infection went away 2 years ago, what are the odds of there still being debris kicking around? That seems to be a leap to me. The debris has to be recently generated. Yeah.
Yes, because there are so many mechanisms by which the body cleans up debris. So if there was debris for a couple weeks after infection and there was no more, but we're talking about people who are still sick a year later, 2 years later, that's just starting to get strange, that the debris is just, and they're not even, these are sometimes patients that are getting sicker So that would be we'd have to start to study debris in a totally new fashion to just understand some sort of new phenomenon there by which someone could actually be as sick as they were 2 years before just due to debris. Yeah.
Yeah, and generally the simplest explanation is the right one. And
Yeah.
when it comes to looking at this, the simplest explanation is actually viral persistence, isn't it?
To me, it is, yes, because it then can start to explain so many of the other things that are measured. So we could explain some of the autoantibody production by persistence of the virus. We can certainly explain how other pathogens may themselves be reactivated. That is definitely, I think, a trend that I haven't described as much, but all of us are seeded with other organisms, other pathogens that are the herpes viruses, so Epstein-Barr, cytomegalovirus, but also sometimes, for example, toxoplasma is a parasite that 1/3 of the world population harbors. In the US, I think 11% of the US population harbors latent toxoplasma, this parasite in the central nervous system. This is just something we know that's rarely discussed. Toxoplasma causes neuropsychiatric symptoms, inflammatory symptoms. So you have this possibility that depending on your unique, what I'm going to call pathogen burden that you acquired up until the point where you got COVID. And again, these pathogens, as long as your immune system is robust and you're in fairly good shape, it's keeping these pathogens in check. It's saying, oh yeah, you're there, but no, you can't, we're going to keep you under control. But then you get COVID and a lot of these other pathogens, their activity can surge up, which could happen with Borrelia. And patients here in the US, they might have gotten bit by a tick and they have Borrelia or Bartonella, but they didn't realize. And it remained in a small state and then that got kicked up. So all of that and those pathogens can also contribute to autoantibody production via mimicry, right? So you have a whole host of the other organisms that can come into that phenomenon. And then you even have, for example, we've mentioned, I'm not sure, there's clotting that's been documented in patients with long COVID. So Rhesia praetorius has documented microclots in patients with long kind of amyloid fibrin clots. And while we do need some more research on the clots, we need to better understand what they're made of and characterize them in a more of a capacity. It still is not surprising that the blood would be clotted because SARS-CoV-2 in number of studies with COVID is now been shown to drive clotting type mechanisms. And so one of the things that you can begin to connect there is that if you don't fully clear the virus and a little bit of spike keeps leaking into the blood, that will keep the blood clotted, which makes sense. And then another thing that can contribute to clotting that actually Rhesia praetorius and her colleague Doug Kell have shown in several papers before COVID ever started is leakage of bacteria from the gut or oral microbiomes. So they've shown in previous papers that lipopolysaccharide, which is a component of the bacterial cell wall that will leak from the gut or these communities when they're inflammatory, that catalyzes clotting. So if you have a person who has a little bit of SARS-CoV-2 left in intestinal tissue, spike is leaking into the blood, bacterial product is starting to leak more into the blood, now you have ongoing clotting issues. All of that starts to tie together to me in the most straightforward fashion where we don't have to go through lengths to come up with theories to figure out one thing. It actually fits together in a pretty concrete way. And one more thing I'll add in is that I'm sure you've seen that patients with long COVID are increasingly getting diagnosed with small fiber neuropathy. So that's a condition where the peripheral nerves, the very small sort of C fiber nerves that are in the periphery of the body begin to become diminished or die off. And patients can be assessed for small fiber neuropathy. They'll just do a punch biopsy. They obtain a piece of the tissue. And at this point in time, if you get the diagnosis, they count the nerves. They say, yeah, there's less nerve here. It's died off. And then you get the diagnosis, but no one's really looking into the question of why is there this die off of nerve? And one thing, if you look through the literature on diabetes, where neuropathy, peripheral neuropathy is fairly common, there is some possibility that if the blood becomes coagulated, it can't reach the little tiny capillaries or blood vessels that feed these peripheral nerves. So these little nerves they demand oxygen, they need blood. So if the blood is clotted, it might not be able to reach those little nerves, which could be a big reason that they die off. So that could be connected then. The blood clotting could be connected to the small fiber neuropathy. And then once the nerve dies off and there's less blood perfusion there, if that's the case, that area becomes in the tissue hypoxic, which might make it a very good site for influx of other organisms, even SARS-CoV-2. So you kind of get this secondary colonization. And we're doing a whole study where we're taking those punch biopsy samples and we're working them up to study all of these trends. That's one of my favorite studies. So again, you can connect these topics through some of those mechanisms.
Hope you found that discussion interesting. Personally, I found it absolutely fascinating. In terms of future research, we may not find the smoking gun in terms of replicating virus and linking it inexorably to persistent symptoms, but we may find efficacy in terms of antiviral treatments. One has to hope we'll get a Paxlovid trial on long haulers soon, and we'd effectively get to kill two birds in one stone that way. Anyway, coming up, I'll be talking to world-leading MCAS expert, Dr. Tanya Dempsey, about why women suffer from long COVID more frequently than men, and just why their symptoms flare in time with their cycles every month. And I'll be checking in with Dr. Sally Riggs with a very convincing answer as to why type A personalities and those with prior trauma do seem to be at higher risk of developing long COVID. Till next time.